Montpellier, France. He's going to tell us about his research on the ancestral synteny of asteroid and rositic clays.
Buenos dias, good morning. I would like to thank the organizers to give me the opportunity to share our results with you today. The work I'm going to expose today was done before several genomes were finished. The only one which was available was the grape genome. This work, as the title indicates, concerns the analysis of the general synteny between genomes from very distantly related species, Coffea, vitis, and tomato. You know that Coffea and tomato belongs to the Rubiaceae and the Solanaceae respectively, and both are asteroids, and vitis is a rosid, and it's also a very early, let's say, it's a very early divergence in the angiosperm general tree. This is a very summarized tree, of course. The divergence between Canephora, let's say between the Rubiaceae and the Solanaceae, is about 85 million years ago, but the divergence with vitis is about 120 million years ago. Just to indicate where these days the natural, I'm speaking about the natural habitat of this free species is. Of course you can find all the free plants now all around the world, but they are originating for vitis in the Caucasus and let's say Persia, Iraq and so on region. Coffea canephora is originating from West Central Africa, and tomato mainly from the Andean regions of Peru, Ecuador, and so on. We did this work using COS. What are COS? COS are conserved autologous sequences. These sequences also are unique in genomes. Why are they called conserved autologous sequences? That means that you can define them in one species, for instance in Arabidopsis, and you can transfer them in other species. You can find them, their equivalent, in other species. So it's what was done, looking for the COS first in Arabidopsis, then transferring them, first work did transfer them in Solanaceae, and transfer them in coffee. And we looked about we looked how many COS that were identified in coffee and tomato were also present in the grape genome. Of course, some criteria were defined, and at the end, we ended up with 867 COS distributed in the genome of these free plants. Of course, when you do comparison 2 by 2, we don't have all this set of COSs available. We had 380 COSs between coffee and tomato, almost 300 between coffee and grapevine, and 470 between tomato and grapevine. We didn't work only with COS alone, isolated COS, because some of these genes may also rise from a duplication and then lose loss of the original paralog. So we worked with blocks, with conserved blocks. We defined a block as containing at least 3 COS, and we mainly focused on these blocks. So what happened? When we compare chromosome by chromosome from coffee, for instance, with the chromosomes of tomato, we looked at all the conserved blocks present on one chromosome of coffee and where were they found in tomato. As you see, we had some situation like this one. Almost all the syntenic blocks that were identified in this chromosome are found on only one chromosome of tomato. Of course, some blocks are also spreading in other chromosomes. Some rearrangement occur. But we had also a figure that showed that there are also much more rearrangements between coffee and tomato. the big part of the synthetic blocks are found on one chromosome, but there is a spread on many different chromosomes. So huge rearrangements in that case. It was the same situation between coffee and grape. I recall you that the divergence time between tomato and coffee is much shorter than the divergence time between coffee and grape. And as you can see, we have the same time of figure between grape and coffee. One coffee chromosome mainly found on, in that case, 2 grape chromosomes, but coffee has only 11 chromosomes, grape has 19 chromosomes, so there is, of course, some breakage, let's say, that is expected. And we had also this situation that means a much wider spread of the synthetic blocks from the coffee chromosome on the grape chromosomes. When we look now at the micro level, that means at the level, in that case, of a back which was fully sequenced. We sequenced the back coming from coffee that contains gene. The ovate gene is interesting. It has an agronomic interest because it intervened. It was first identified in tomato. It intervened on the shape of the fruit of the tomato. So it may also play a role maybe in coffee. So that's why we choose this back to sequence. It's a back which is very rich in genes. It's not very long, 174 kb, and it has 23 putative genes. These genes were identified thanks to transcripts that were perfectly matching the sequence of the back. Now when we look at the comparison between this back and not only the presence of the gene, but also the orientation of the genes, that means we have synteny and we have collinearity in that case. Synteny is just the linkage of the sequences. Collinearity, that means we keep exactly the same order and the same orientation of the genes. And in that case, you see that we have 19 different genes that are common between coffee and grape. What, of course, sometimes they are here, for instance, just local duplication. And sometimes there are also deletions or maybe insertions. We don't know if it is deletion in grape or an insertion in coffee. What happens when we compare with solanaceae? We didn't compare only with tomato, we also compared with different solanaceae for which the sequence was available, the equivalent sequence of the region was available. I recall you that the tomato genome was not available when we did this work. But as you can see, unfortunately we didn't have the full sequence to compare between the different solanaceae and coffee and grape. But if we focus, let's say, on this region, from here to the end, for tomato we have much more rearrangement and a less well-conserved synteny between solanaceae and coffee or grape. We have even local inversions in the solanaceae, local duplications, inversions between the solanaceae. That means the solanaceae, it seems that everything indicates that the solanaceae underwent an important rearrangement after the separation, let's say after the divergence from the rubiaceae. And if you recall the talk, Philippe's talk, there was, of course, in the solanaceae this possible event of global duplication, which, of course, implicates a huge amount of rearrangements in the chromosomes further. So, if we look, let's say, at the interest of the synteny, we can see that when you have a gene of interest, which is, let's say, proven to be interesting in one species, if you have the possibility to follow this gene with COS markers, you can transfer the COS markers from the species you know here, in the case of tomato, for instance, and you can look at the presence of your gene of interest in the species which you are working with, which is your preferred species. So there is an interest, an agronomic interest of this work of Synteny, because you just then focus in a program, for instance, of marker-assisted selection, you just focus on that region with the markers bordering your gene of interest, if you have, of course, those costs available. So it's a very quickly shortcut. You don't need to map your, of course you need a basic map, but you just focus on the region of your gene of interest, bordered by the cost marker that you can easily transfer from one species to another. So, in conclusion, we found a significant conservation of Synteny between distantly related species and very distant species, and it's the first work that, let's say, involved very distantly species from the asteroid and from the erosive clades. And this conservation is found both at the macro and the micro level. And as I showed you, maybe it's a particular case of the region we sequenced, but the conservation at the micro level and conservation of Synteny and collinearity is better between grape and tomato, that means between the distantly related, the highly distantly related species, than between the Solanaceae and Rubiaceae, more closely related species. At the Ovate locus, the Synteny and collinearity conservation did not decline with increasing phylogenetic distances. Exactly what I just said. The Synteny conservation, both at the micro and macro level between distantly plants, suggest that the time factor alone does not explain divergences. And here I have to recall you, tomato and all the Solanaceae here analyzed are annual plants. Coffee and grape are perennial plants. So maybe time alone is not the only factor, but the generation time also has to be let's say, taken under account. And the synthetic studies between supposedly remote species are useful for facilitating the isolation of important genes of agronomy and to be used in marker-assisted selection breeding programs. So I just want to thank the IRD and Nestlé for supporting this work. All the data, all the costs that were identified to do this work, and all the mapping work is available on the MocaDB database, and there is a poster that describes the database. It's a database maintained at IRD, but which is publicly available, open to every one of you working on breeding or whatever concerning genetic of coffee. And of course the work was published this year in BMC Genomics. And I thank you very much for your attention.
Thank you very much, Alex. We have probably time for one or two very short questions, if anybody in the audience have any questions for Alex. Okay, everybody understood everything well. Thank you, Alex.